Hello everyone and a warm welcome to Heredity Bioacademy YouTube channel. I am thrilled to have you join us today for an exciting study into the fascinating world of microbiology. Our focus center on the test that expose the metabolic process of microorganisms, the sulfur reduction test. So buckle up for an insightful journey into the captivating topic. Don't forget to hit that subscribe button and stay tuned to more enriching contents. Let's join into the sulfur reduction test. Continuing to the introductions, microbiology that shows the metabolic ability of certain microorganisms. Imagine a microscopic world where elemental sulfur and sulfur-containing compounds become sources of metabolic energy for specific microbial inhabitants. These remarkable entitles are suitable like sulfur-reducing microorganisms, showcasing an extraordinary ability to metabolically reduce sulfur, both organic and inorganic, to harvest essential energy. In-house within the kingdom of microbiology are varied microorganisms including Archaea, aerobic, facultative, and anaerobic bacterial species, each equipped with a unique capability to convert sulfur into hydrogen sulfide. This metabolic feat serves as a testament to the microbial world's intricate and various energy generating mechanisms. Now, the sulfur reducing ability isn't a universal trait among all microorganisms. Therefore, scientists and microbiologists have devised a clear microbiological test known as the sulfur reduction test. This test becomes a diagnosis tool allowing us to distinguish the microbial players able with the distinctive talent. But you may wonder, how does this test work? During the sulfur reduction test, microorganisms are presented with a sulfur-containing compounds. And if they possess the ability to reduce sulfur, the byproduct is none other than hydrogen sulfide gas. This gas, an informative sign of sulfur reductions, is reduced by an indicator during the test. Hence, the test is also suitably called the hydrogen sulfide test. The significance of this test extends beyond the laboratory walls. It emerges as a crucial diagnosis tool in both medical and non-medical microbiology laboratories. In the water analysis, it becomes a sentinel diligently detecting the presence of coliforms. In the identification of fatal pathogens and the differentiation of enteric pathogenic bacteria, the sulfur reduction test takes center stage. Moreover, it becomes a suitable tool in characterizing bacterial isolates, distinguishing between pathogenic and non-pathogenic strains. Now, we will move on the objective of sulfur reduction test. First one, to assess the ability of bacteria to reduce sulfur compounds. Second one, to detect the ability of bacteria to produce hydrogen sulfide gas. Third one, to biochemically differentiate bacteria for presumptive identification. Now, we will move on the principle of sulfur reduction test. Sulfur reducing bacteria can metabolize sulfur-containing compounds present in the culture medium and reduce the sulfur to hydrogen sulfide. The released hydrogen sulfide react with the indicator, mostly ferric ions or lead acid, to form black color insoluble compounds, which turn the whole medium black. Thus, the development of black color in the medium after the incubation period indicate that the test organism is capable of reducing sulfur. Now, we will discuss for this what are the requirements for sulfur reduction test. In this test, we require for some of the culture mediums like sulfide indole motility mediums, Kliger's iron agar, triple sugar iron agar mediums, lead acid agar are commonly used for this test. Among these, the sulfide indole motility medium is the commonly preferred mediums to perform this test. In these mediums containing sodium thiosulfide as a sulfide source and peptonized iron as an indicator. Now, preparation of sulfide indoor motility medium. Measure the appropriate amount of seam agar powder and mix it with the appropriate volume of distilled water in a conical flask. Then stir well using a magnetic stir or manually you will do it and heat to boil so that the agar dissolves completely in the water. Dispense about 5 ml of the medium in each test tube and loosely put on the cap or cutter block in the opening. After that, autoclave the tubes at around 121 degrees centigrade and 15 W pressure for 15 minutes. No additional reagents are required while using seam or other sulfide containing agar, but lead acetate paper is needed while performing the lead acetate paper method. Some of the equipments required in this test like test tubes, incubator, weighing machines, inoculating wires, Bunsen burner, autoclaves. Now we will discuss for the procedure of sulfur reduction test. First one, seam agar method, that is called tube method. Utilize a sterile inoculating loop to collect colonies from a fresh 18 to 24 hours of bacterial culture. Insert the wire halfway into the seam medium tubes 3 to 5 mm above the base. Inoculate the tube aerobically with a loose cap at 37 degree for approximately 24 hours. Next day, monitor the medium for a emergence of black colorations. Next method is that lead acetate paper method. Using a sterile inoculating loop, just several colonies of sample bacteria from fresh 18 to 24 hour culture and inoculate the nutrient growth or peptide water medium. Position a lead acetate paper strip so that one end hangs just over the medium and other end is secured at the tube's neck with a cotton block or screw cap. Inoculate the tube aerobically at around 37 degrees centigrade and observe the blackening of the paper strip after 24 hours. Now, the result and interpretation of sulfur reduction test. A positive result is indicated by blackening the medium and turning the lead acetate paper into a black color. A negative result is indicated by no blackening of the medium or no change in the color of the lead acetate paper. This is the picture represented for the result. Sulfur reduction test result for common bacteria, providing crucial understanding into their metabolic capabilities and aiding in microbiological differentiation. Understanding these outcomes enhance our comprehension of microbial behavior and contribute to various fields of microbiology. Highlighting table result for sulfur reductions and non-reduction bacteria, offering a comprehensive view of their distinct metabolic characteristics in microbiology. Thanks for watching this video. More informative video, kindly like and subscribe to the channel. Thank you.